Dear students, in this video, I'll be explaining about substrate strain theory with regard to the enzyme lysozyme. When a substrate binds to the active site of the enzyme, there will be conformation change in the enzyme, and this induces a pressure or strain to the substrate, leading to the formation of the product. So this concept of substrate strain theory explains the role of enzyme in increasing the rate of the reaction. In fact, it is the combination of both induced fit model as well as the substrate strain which are considered to be operative in the enzymatic action. So now let us know about the enzyme lysozyme. So lysozyme is also called as muramidase, or it is also known as N-acetyl muramyl hydrolase. The enzyme commission number is 3.2.1.17. So this enzyme belongs to the class 3, nothing but the hydrolase enzyme. So this enzyme makes use of water molecule in cleaving the bonds present between the residues. So it's a cationic protein of 14 kilodalton and is of 129 amino acid residue occurring in animals, plants, bacteria, and viruses, found in neutrophils, macrophages, serums, saliva, milk, honey, and hen egg white. So this enzyme was discovered by Scottish physician and microbiologist Sir Alexander Fleming in 1921. So this enzyme catalyzes the breakdown of peptidoglycan layer found in the bacterial cell wall. So what it does, it acts on or attacks the rigid layer, which is a protective layer present in the cell wall of bacteria. It is called as muramin, peptidoglycan or muramin. It acts on it and breaks the linkages present between the residues known as N-acetyl glutamic acid and N-acetyl muramic acid. So these residues make up the peptidoglycan layer. Peptidoglycan is nothing but the combination of protein and carbohydrate. So this strand is acted upon by the lysozyme enzyme. So this is a natural substrate for the lysozyme enzyme. It is antibacterial in nature, so it acts on it and cleaves it. So this strands, polysaccharide strands, these are cross-linked through short peptide bridges at the lactyl groups of the muramic acid residues. So here you can see that these are the strands containing alternate residues of NAG and NAM like this. And these are linked by peptide cross links. So these are the polysaccharide chains. Therefore, it is made up of both protein as well as the carbohydrate. So the strands the bonds present between NAG and NAM is 1,4-beta linkage. So the enzyme acts on 1,4-beta linkage and it cleaves the strands into fragments. Now let us see how the lysozyme acts on peptidoglycan layer. So this is the substrate. Here only 2 residues I have taken as an example to explain the action of enzyme on the substrate. So lysozyme is a part of innate immune system and is antibacterial in nature. When a bacterium enters into our body, the lysozyme counteracts it. So the substrate fits into the active site of the enzyme. About 6 subunits of this substrate fits into the active site of the enzyme. When it fits into the active site, there is a conformational change in the enzyme. So when there is conformational change in the enzyme, it strains the substrate. So there is change in the conformation of the substrate. As you can see here, the NAG has residue here at the top, and NAM has the R group. This R group on the top in the NAG and R group in NAM at the bottom. So when this fits into the active site of the enzyme, here there is strained conformation of the substrate. So there is transition state of this substrate, and you can see R is towards the inferior side, or towards the bottom. So there is change in the conformation of the substrate. So here the substrate is strained. So when this substrate is strained, now the enzyme reaction starts. So here induced fit also happens, and also substrate strain as well. So here 2 amino acids, glutamate and aspartate, these 2 amino acids which are present in the active site, they take part in the hydrolysis reaction. Glutamate, at first, it donates its proton to the NAM, and aspartate, it attacks the C1 carbon of NAG, and it forms a transient covalent bond with the NAG. Here, glutamate, after donating hydrogen, it loses hydrogen, and it will be starving for a proton here. Now there is a bond present between, covalent bond, transient, that is the temporary bond present between the carbon and the aspartate. In the next phase, hydrolysis takes place. So water molecule comes into the picture. The hydrogen is taken away by the glutamate, and the oxygen atom attacks here the carbon. So this is not the right structure. Here the oxygen attacks the carbon atom of this, and it breaks the transient covalent bond that is formed between the aspartate and the carbon, and this leads to the formation of a bond between the OH group here. So the glutamate and aspartate, they regain their original structure, and here fragments are formed. The product is formed. Finally, the products and the enzyme dissociate, and this is how the lysozyme act on the peptidoglycan layer. So lysozyme, it has dual activities. It acts as a lytic enzyme, and it lyses the cell wall of the bacteria, and also it activates the autolytic enzymes present in the bacterial cell wall. And this lysozyme, it is used in treatment of periodontitis, and also it is administered in chewing gum, and it is implemented to prevent tooth decay. So it is also used as an analgesic drug to treat the patients who are suffering from cancer. And it's been used as potentiating agent in antibiotic therapy. Thank you.